to survive. So let's look at, I'm going to draw just kind of an overview of this. So here's a big old mitochondria, and then we have the inner membrane. And so what we have here out in the cytosol, as Maria said, is we have the first step, which is glycolysis. So glycolysis, it literally means the splitting of sugar, and lysis means the splitting. And so you are taking one glucose molecule and converting it to two pyruvate molecules. And so remember, you're taking a larger molecule and breaking it down into 2-3 carbon smaller molecules. And so so while this process, if you watched the video yesterday, is a series of reactions. So there are many reactions that take place in between here and here. In those series of reactions, we get a net gain of ATP. You get some ATP out of this. This is kind of a bonus here. The 2 ATP that is the net gain of this is not a lot of ATP. It's not enough to keep your cells alive. And so therefore, this is not the main purpose of glycolysis. Can somebody tell me what the main purpose of glycolysis is?
To make the pyruvate?
To make pyruvate, which is, so that is definitely one of its main things, but when it does from glucose to pyruvate, it makes something that is going to be helpful later on in this process. Sage?
NADH for the electron transport chain.
Yeah, NADH for the electron transport chain. So let me talk about where the electron transport chain is. So this whole process, this arrow here represents a lot of things. All right, so glycolysis is the first set of reactions. Then, as we go into the mitochondria, the second step is called the citric acid cycle. And it's called a cycle because what starts, the molecule that starts at the beginning of the cycle goes through a series of reactions and you end up with that same molecule. So it cycles around and around. And so that's why it's called a cycle. And then the third step includes the electron transport chain, but the third step has a special name that starts with the O. Didn't I remember? Big words. Do you remember what that? OX? Close. Oxidative phosphorylation. So what is oxidative phosphorylation? There's two things that are a part of it. The electron transport chain, which I'll write out now, but from now on I'm going to always abbreviate that as ETC. The electron transport chain and this process called chemiosmosis. That's your third step. This is where most of your ATP is made. All right, so the purpose of glycolysis is not to make ATP. The purpose of glycolysis, as Sage said, is to make NADHs. Now, we talked on Tuesday about NADHs. We start out with a molecule called NAD+, and NAD+ reacts with 2 hydrogen. Where do these hydrogen come from? They come from the hydrogen that's in glucose here. So here's glucose, your 6 carbon molecule. This is the hydrogen. All right, and so 2 hydrogens come off of glucose, so NAD+ plus 2 H's gives you, at the end, NADH plus an extra H+. So real quickly here, remember your 2 hydrogens have 1 proton and 1 electron spinning around the nucleus, and then, so we have 2 of those, 1 electron. So one of these electrons, different color here, one of these electrons, notice, goes to NAD+. The NAD+ has a positive charge because it lost an electron. And so this electron here goes to neutralize that positive charge, and so therefore you don't have a positive charge on this. But when this gives off an electron, it becomes H+. And then we have an extra hydrogen, proton and electron. That's the hydrogen here. So this is hydrogen right here. So therefore NADH has 2 electrons that were part of hydrogen that were once part of glucose molecules. And so this carries 2 electrons that was once part of glucose. The H plus is the H plus left over here. All right, so this is the carrier NADH. So I'm going to draw here an arrow from this reaction here. What comes out over here and comes over here and is used in the electron transport chain here is this NADH. This carries to the ATC. All right, that's what that does. And that's the purpose of glycolysis, to begin to oxidize glucose. What does oxidize mean? Losing or gaining?
Losing.
Losing electrons. So therefore the electrons are being lost from the hydrogen that was once part of glucose and being carried to the last stage, the electron transport. All right? And so where we're going to start today is what happens to these pyruvates and how we get to the next step, which is the citric acid cycle. So this is the third section. So before the citric acid cycle can begin, pyruvate must be converted to acetyl-CoA, which links the cycle to glycolysis. So what does this mean? So your book and all textbooks say that there's 3 main stages for cellular respiration, and there are. These are the 3 main stages, but it looks like at the end here, pyruvate should go and enter into the citric acid cycle, but that's not actually what happens. So what happens is the pyruvate enters into the mitochondria, but the pyruvate are converted to 2 molecules called acetyl-CoA. And those are what enter the citric acid cycle. So something happens to these molecules to be converted to acetyl-CoA and enter the citric acid cycle. And that something is represented in this picture. So what are we looking at? This picture shows you the actual picture of pyruvate. This right here is the double membrane of the mitochondria, just like I have here. So it's entering into the mitochondria and being converted to acetyl-CoA, which will enter the citric acid cycle. So let's look at what happens here. So pyruvate, this part right here, looks an awful lot like what functional group that we've learned about?
Carboxyl.
Yeah, this is the carboxyl group. Carboxyl groups make the molecule what? Acidic, right? And if it's acidic, it gives off what in solution? H plus. So notice here that we have the C with a double bond and O and the O with a negative charge. What does that mean? It means that this has already acted as an acid by giving off H pluses. So it's already acted as an acid. So sometimes when we talk about the end of glycolysis in pyruvate, sometimes you'll see it written, instead of glucose being broken down into 2 pyruvates, sometimes you'll see it written as pyruvic acid. And these guys here, it's the same molecule, it's just whether or not it's already acted as an acid. So pyruvic acid would look like this. It would look like, this would not have a negative charge, and you'd have your OH there. All right? And so that's what the acid looks like, pyruvic acid. If it acts as an acid and gives off its H plus, it now looks like this, and it's called pyruvate. So just so if you see pyruvate, the word pyruvic acid, or pyruvate, and say, okay, glycolysis makes 2 pyruvic acids. I thought you said pyruvate. That's why. All right? And so, so these guys enter. Notice here, the mitochondria, there's a transport protein that allows it to enter. And so then it's going to go through a couple of things, 1, 2, 3 steps, in order to make your acetyl-CoA. So let's look at what those 3 steps are. CO2 is released. So CO2 is this part right here. So it was once the carboxyl group. It's going to leave as a molecule of carbon dioxide. What happens to this? We breathe it out. So this is waste that we breathe out. All right, second step is we make some more NADHs. So notice here, a lot of people always get confused about these arrows that look like this. What this means is, see, this is a series of reactions here. We just don't need to know all of them and the logistics of it all. Just the big picture here. So what this means is NAD plus goes into the reaction and comes out as NADH. How did it become NADH? NAD plus goes in, it took 2 hydrogens off of those intermediates and became NADH. So this is an electron carrier, so it carries, just like all the other NADH, just 2 electrons, and it's going to go to the electron transport chain. So that goes in and goes out of this reaction here. So we get some NADHs made. Then we have this molecule coenzyme A. This is a molecule just found in the cell. That's all we need to know. Molecule in the cell. And so this attaches to it. So notice here the acetyl-CoA. This part right here is this part right here. All right, and so that's the CoA part, the coenzyme A. And notice that really the big difference here, pyruvate and acetyl-CoA, is what's attached to this carbon. C3, CH3, C with the double bond oxygen, that's the same. It's this part that's different. All right, and so what we get here is your molecule of acetyl-CoA. So for one glucose molecule, how many pyruvates do we get? 2, right? So this is showing you 1 pyruvate making 1 acetyl-CoA, so this process happens 2 times. So realistically, there's 2 pyruvates that enter. We can double this, 2 pyruvates that enter, which means then we could put a 2 in front of everything. So 2 NADHs are made, 2 CO2s are made, 2 coenzyme A's are needed to make 2 acetyl-CoAs at the end, okay? And so, so that's what we have here, so the 2 acetyl-CoAs. So let's write here, I'm going to add this to my picture here, the blue is the step here. So we do get some NADHs made. So and those NADHs are going to carry electrons to the electron transport chain. So NADHs. So we further oxidize or taken electrons off of molecules that used to be part of, or used to be glucose. And so in this process of making the 2 acetyl-CoAs, we also get some waste products, and so the 2 CO2s are the waste products. Okay, and so that's, as I said, is waste, and that will be exhaled. And this step doesn't really have a name, and so sometimes they'll call it the intermediate step because it's intermediate between glycolysis and citric acid cycle. Sometimes they'll call it the link reaction because it links glycolysis to the citric acid cycle, and so on. So it doesn't have an official name. They don't consider this actually a main part of cellular respiration because no ATP is made here. But it's an essential part because you can't do the citric acid cycle unless it happens. So we can call this, like this is step 1, this is step 2, we can call this step 1.5, all right? Meaning that it has to happen in order to get to that citric acid cycle, that second step, all right? So that's what that is. All right, so let's look at what happens to the citric in the citric acid cycle. Citric acid cycle is also called the Krebs cycle, from the scientists who discovered it. And it takes place within the mitochondrial matrix. If I drew a little mitochondria here, where's the matrix in the mitochondria? Where would I find that? It's not the folds. What did you say?
Outside.
It's inside the fold. So this part right here is the very interior. This is the matrix. So here we have, so what you said. Oh, that's 2 men. Okay. And so, so it's here in the matrix. And so that's where the Krebs cycle occurs. So what this cycle does, it oxidizes organic fuel derived from pyruvate, generating 1 ATP, 3 NADHs, and 1 FADH2 per turn.
Would that be double then? Okay.
All right, so oxidizes organic fuel. Oxidizes means if the organic fuel is oxidized, the organic fuel is losing electrons, right? Oxidizes organic fuel. So what is the fuel? What enters the citric acid cycle? It's acetyl-CoA. All right, the acetyl-CoA was once pyruvate, which was once part of glucose. And so what we're doing is further taking electrons off of these molecules that were once part of glucose. So we're further oxidizing them. and this is what we get, all right? And so we get a little bit of ATP, some NADH, and a new molecule here, FADH2. FADH2 we're going to see is very similar to NADH. This is also an electron carrier. Notice that it says per turn. Turn is one cycle, one round of this. How many rounds does it take? How many turns does it take for one glucose molecule? One glucose molecule gets converted to pyruvate, which gets converted to acetyl-CoAs. Each acetyl-CoA goes in separately, so therefore we have 2 turns. All right. So I'm going to put here 2 per glucose. So this next picture gives you an overview of what happens in the citric acid cycle. So up until this point here, so in the picture, up into the acetyl-CoA, all of this up here is the intermediate step, so, or the link reaction. And so this is all what I just put here in blue. Pyruvate gets converted to 2 acetyl-CoAs. And so that's what we have here, an acetyl-CoA now going to enter the citric acid cycle. So what happens is the citric acid cycle is a series of reactions. So like there's a molecule, I'll call it molecule X, that when acetyl-CoA comes in, it reacts with it and forms molecule Y. Then another reaction occurs and it forms molecule Z. And we go through a series of reactions until eventually, at the end of the citric acid cycle, you're back with molecule X, and that molecule can greet and react with another acetyl-CoA, and the cycle keeps on happening. So this is why it's a cycle, because you start out with one molecule, and in the end, you end up with that same molecule. So what happens in this series of reactions? We don't need to know all the intermediates, all right, and the names of all these intermediate molecules and so on. We just need to know the big picture. So we see here it loses the CoA part and goes through this series of reactions. And what do we gain as a result of these series of reactions? So we see here 2 CO2s leave. This is waste, right? So this is what's exhaled. Again, so there's some of our carbon dioxide. And here we have 3 NADHs. Now notice here this arrow again going in and out, because in order to make NADH we need NAD+. And so where did the hydrogen come from? It took some of the hydrogens from the intermediate molecules in this reaction to form NADH. So therefore this is going to carries 2 electrons where?
Electron transport chain.
Electron transport chain. I'll just put ETC. Okay, so then we go through some more reactions and we get a little bit of ATP made. So here's our next thing that's made. How do we make ATP? We have to put a phosphate back onto ADP to make ATP. So when it shows you here going in, that's what those are the things that are needed in order to get this out. So then we gain some ATP. And then here we have a second molecule, the FADH2, which we wrote earlier is it carries electrons to the electron transport chain. So how does it do that? Notice it's first a molecule of FAD, and then it picks up 2 hydrogens and becomes FADH2. So the reaction is this. So this is FAD plus 2 hydrogens. So it's just like NAD+, all right, NAD+, plus 2 hydrogens. But it's a little bit easier because FAD doesn't have a charge on it. It's neutral. NAD+ had a charge, so one of the electrons had to go to neutralize it. This doesn't have to happen here. So FAD comes into contact with 2 H's, and it becomes FADH2. It's just that simple. All right, and so it's carrying 2 hydrogens. These hydrogens came off of a molecule. This is from a molecule that once was glucose. All right, so the hydrogen was glucose, was part of glucose. So even though glucose was broken down way here at the first step, those hydrogens and their electrons are cycling around in this citric acid cycle. And so, so therefore, how many electrons is this carrying? It has carrying two hydrogen atoms that were once part of glucose. Each hydrogen has how many electrons? Each hydrogen has one electron, right? So therefore, when we say it's carrying electrons to the electron transport chain, it's carrying two hydrogens, so therefore 2 electrons. So it's carrying 2 electrons to the electron transport chain. So this is another electron carrier. And then we go back, and then that's our overview of the citric acid cycle. So what I'm going to have you do, I'm going to put here, let's turn, I'm going to have you total some things. Total for glucose. What I want you to do is write the total amounts of each one of these things. How much CO2 is made, how much ATP is made, how much NADH is made, and how much FADH2 is made out of the citric acid, just for the citric acid cycle, per glucose. So per glucose molecule, so going back here, 1 glucose going in, right, it's converted to 2 pyruvate and then to 2 acetyl-CoAs. So when we look at this cycle here, this is for 1 acetyl-CoA going in here, so there's only 1 here. But per glucose molecule, we have 2 acetyl-CoAs, so since this citric acid cycle happens twice, we can double all of the numbers. And so, therefore, how much CO2 should we have? 4. How much ATP? 2. How much NADH? 6. And FADH2? 2. All right, absolutely. So, if we relate this back over to here then, for the citric acid cycle, so we get some ATP, so I'm going to put out here, so we get 2 ATPs. Again, not a lot of ATP, so the purpose of the citric acid cycle is really not to make ATP. We get some waste products, the CO2, 4 CO2, which is a waste product. Notice here, 4 and 2 makes 6. 6 CO2s that are a waste product coming out. All right, and so, so then, but these two are not the purpose of the citric acid cycle. The purpose of the citric acid cycle is to further strip electrons or oxidize the molecules that used to be glucose. So take more electrons off of it, and those electrons are going to now be carried again to that 3rd step via 2 electron carriers, NADH and FADH2. So right now, before we get to this 3rd step, glucose is broken down as much as it's going to be. So what happens, let's look at where the parts of glucose went. The CO2 that is leaves is made from the carbons and the oxygens that were once part of glucose. And so the hydrogen part of glucose and their electrons are being carried to the electron transport chain through NADH and FADH2. And so we've all, to this point, we're done breaking down glucose. Now all the energy that was once in glucose is now in these electron carriers that are going to carry the electrons to this third step and use that energy to make the most of the ATP that we make. So most of the ATP is from this third step. And so that's the purpose of the citric acid cycle. All right? So everything up to oxidative phosphorylation is just to make these electron carriers. Okay? Alrighty. So let's go on and look at how that happens. So the citric acid cycle has 8 steps. Now you don't have to know all these 8 steps or anything like that. I just wanted to include this to show you how it's a cycle. Each step is catalyzed by a specific enzyme. The acetyl group of acetyl-CoA joins the cycle by combining with a molecule called oxaloacetate, forming a molecule called citrate. Another way to say citrate is it's citric acid that lost its, or gave up its H+, and already acted as an acid. Hence the name citric acid cycle. It's the first molecule that's made in the cycle. And then you have 7 other steps that eventually decompose citrate back to our first molecule, which is oxaloacetate, making the process a cycle. So that's all I want you to see from this, this picture that we're going to look at next. And then, as we just wrote on the board, the main purpose is the NADH and FADH2 produced by the cycle relay the electrons extracted from molecules that used to be part of your glucose from your food to the electron transport chain. So you don't have to know this step. This picture here is the details of this picture. So this picture is just not showing you the details, it's just giving you the big picture. That's what you need to know. I just included this one so that you could see how it's a cycle. So if we look here, here's the cycle, here's the acetyl-CoA coming in, and here's the oxaloacetate. These guys react to form citrate or citric acid, hence the name, citric acid cycle. It's the first molecule that's made. We get our 3, here's the NADH is made, here's the FADH2, the ATP, carbon dioxide's given off, and so on. So it's the same picture as before. I just wanted you to see that there's a bunch of reactions, and eventually you end up with the same molecule that then can meet with the next acetyl-CoA, and this keeps on going on and on and on. All right? And so that's the citric acid cycle. So let's look at then this last step, oxidative phosphorylation, because so far we haven't made enough ATP to keep our cells alive. So this next step needs to happen. So following glycolysis in the citric acid cycle, NADH and FADH2 account for most of the energy that's extracted from the food. I just talked about that. These 2 electron carriers donate electrons to the electron transport chain, which is going to power ATP synthesis and the making of ATP via oxidative phosphorylation. We'll talk more about oxidative phosphorylation in just a second. So, electron transport chain, what is it here? The electron transport chain is in the cristae of the mitochondria. Most of the chain's components are proteins, which exist in multi-protein complexes. The carriers alternate reduced and oxidized states as they accept and donate electrons. And then these electrons drop in free energy as they go down the chain and are finally passed to oxygen. So let's look at some of the meanings of this. All right, so electron transport chain is in the cristae of the mitochondria. So where's the cristae of the mitochondria?
The folds.
These are the folds. And so the electron transport chain, it says, are proteins which exist in multi-protein complexes. So remember when we looked at cell membranes, they're lipid bilayers with proteins embedded in them. And so the parts of the electron transport chain and the proteins in the electron transport chain are in the folds of the membrane. So if I were to draw that here, on here, you'd have these complexes within the cristae. And so the electron transport chain is going to, electrons are going to move from molecule to molecule in these proteins in the inner membrane of the mitochondria. All right? The carriers alternate reduced and oxidized states as they accept and donate electrons. The reduced state, are they going to be accepting or donating? Accepting. So these guys accept electrons. And oxidized, they're going to be donating. All right, according to this. All right, and then they drop in free energy as they go down the chain and are finally passed on to oxygen. So here in comes the role of oxygen in cellular respiration. We haven't even talked about that yet. So we have to breathe in oxygen. It has to do with the electron transport chain. So let's look at this picture here. Whoops. Let's look at this picture here. So what am I looking at here? This is the electron transport chain. Now, in the bullet earlier, we said that most of the chain's components are proteins, and then they exist in multi-protein complexes. That's what we're looking at here. So each one of these oval-shaped guys right here is a protein that's part of the electron transport chain. But notice that we have these complex proteins here. This complex that this has made out of two smaller proteins. This one's not in a big complex. Here's complex 2. All right, there's 2 of them here. Here's complex 3 that has 3 of the molecules of the electron transport chain. This one's not in one, and here's 4. So that's what that means. And so what happens in the electron transport chain is all these electron carriers, NADH and FADH2, bring their electrons to this third step. So what does that mean? So each NADH, so at the top here you have NADH and you have an arrow here. How many electrons did NADH carry? 2, right? So I'm going to write here it's dropping off 2 electrons. Okay? 2 electrons. So now this molecule has accepted electrons. It's been what then? Oxidized or reduced? It's reduced, all right? Reduction is gaining electrons, so it's been reduced. So now this molecule is going to pass the electrons to this molecule. Let me ask you, why in the world that would happen? Why don't the electrons just stay with that FMN molecule? What do you think it has to do with?
Because the FES molecule is more electronegative, so it takes the electrons from it.
Absolutely. So as we go from down here from molecule to molecule, so our trend here, as we go to from molecule to molecule, is we have increasing electronegativity, which means that it draws electrons towards itself. And so those electrons now get passed to FES. Now FES has now become reduced or oxidized? Reduced. And when FMN passes its electrons to FES, FMN now is oxidized. Then this one passes its electrons here. This one becomes reduced while this one becomes oxidized. And so what happens is this is a series of redox reactions where one gets reduced and then it gets oxidized as it passes the electrons to the next one, and so on and so forth. Redox reactions release energy. And so therefore, as these electrons are passed from molecule to molecule in the electron transport chain, it releases a little bit of energy at a time, and that energy is what's going to be used to make ATP and to make the most ATP in this end third step here. So when we get to the end of the electron transport chain, where do the electrons go? Notice here, there's an arrow here. So now our 2 electrons have been passed from molecule to molecule, releasing energy to make ATP. So notice here that the 2 electrons meet up with, it says, a half of an O2 molecule. O2 is oxygen double bonded with another oxygen. So a half of an O2 molecule is just in one oxygen, right? And so, and then it says two H pluses. Well, what are the two H pluses? What is an H plus? It's a hydrogen atom that has lost its electron. So it has an H plus as one proton, but it's missing its electron that's supposed to be spinning around here. So this is an H plus, and then we have two of those. And so what happens is the electron is missing here. So we have two H pluses. Notice though that electron, there's two of them here. So I'm going to color code this. So these two electrons right here come and replace the missing ones. So now instead of two H pluses, now we have a nice whole H atom. Two H's and one O make one water molecule. All right, and so this is your water molecule. And so water, this is the water that's released here at the end. So the oxygen, what's the role of the oxygen? So the oxygen is what we call the final electron acceptor. Because it's the last thing that the electrons going down the electron transport chain are going to meet up with. Why do the electrons go to oxygen? Because it's the most, yeah, this is the most electronegative out of all of them. And so the oxygen is really important to draw the electrons through the electron transport chain, reducing or releasing energy to make ATP. All right. Without the oxygen, there wouldn't be drawn as much through the electron transport chain, which would reduce the amount of ATP would be made. All right. So that we talked about NADH, but let's talk a minute about this other one, FADH2. FADH2 is a second electron carrier. FADH2 is only made where? In what step? FADH2 is only made in the citric acid cycle. It's not made anywhere else. So I'm going to write here that FADH2 is from, I'm just going to put CA cycle, citric acid cycle. And I'm also going to put that FADH2, that from this less ATP is made than from NADH. Why do you think less ATP is made from FADH2 versus NADH? It has two hydrogens, so it's also when we draw, let's do this arrow here. When we go to this arrow down here, it's also dropping off two electrons, just like NADH. But look at where it's dropping the electrons off. Is it at the beginning of the electron transport chain? No. And so remember that the energy comes from the redox reactions. That becomes reduced, then as it gives it away, it becomes oxidized, and the next one becomes reduced. Then as they give electrons away, this one becomes reduced, and this one becomes oxidized. Since this starts later on in the electron transport chain, there's less redox reactions, less energy, and therefore can make less ATP. So at FADH2, when it drops off its electrons, and the electrons move down the electron transport chain starting here, makes less energy than if we start at the very beginning. All right, but they both can make ATP, and the energy can be used to make ATP. It's just a little bit less. All right, so that's your electron transport chain. Have we talked about has any ATP been made? No, we haven't made any ATP yet. We just talked about the electrons moving through the electron transport chain to release energy to make ATP. So we need to look at how does that actually make ATP. So your next bullet says the electron transport chain generates no ATP. So what is the electron transport chain's purpose? Function is to break the large free energy drop from food to oxygen into smaller steps that release energy in manageable amounts. So what does this mean? If we go back to this picture here, when on Tuesday I talked about how in the lab, if you were to take hydrogen and oxygen and make them combine to form water, it would release, it's very explosive, it would release lots of energy in the form of heat and light and so on. And so, so that would mean there's not a lot of usable energy there. So what happens is that in our glucose here, we slowly break it down, get the electrons from the glucose molecule, and then drop those electrons off in this electron transport chain, which goes from molecule to molecule, releasing small bits of energy. That small bits of energy is available to do work. And so eventually, by the time we get to making water, a lot of that energy has been converted into a type of energy that can be used to do work. So it's not as wasteful. And so that's what it means here, into smaller steps that release energy in manageable amounts. Kind of like us burning gasoline. I used that example on Tuesday, burning gasoline in our car. We could throw a match on the gasoline and get a lot of energy out of it, or we can burn it in a more controlled way, and we can get it to do more work for us, and that's what we do with gasoline in our car. All right, so how do we make the ATP? This is the process called chemiosmosis. Electron transfer in the electron transport chain causes proteins to pump H pluses from the mitochondrial matrix to the intermembrane space. Then those H pluses move back across the membrane, passing through channels in ATP synthase. ATP synthase uses the exergonic flow of H pluses to drive the phosphorylation of ATP. This is an example of what we call chemiosmosis, the use of energy in an H plus gradient to drive cellular work. So let's break this down here. So let's look at the first bullet. Electron transfer in the electron transport chain, so the electrons moving down the electron transport chain, causes proteins to pump H pluses. What does that mean?
Active.
This is active transport. What does that mean?
Uses energy.
Uses energy, right? And in concentration, we usually go from low to high. So low H plus concentration to high H plus concentration. That's what that would mean. So then it's pumping H pluses, now let's look here, from the mitochondrial matrix. So where do we say the matrix was?
On the inside.
In the inside here. So here's the matrix. And it says pump from the matrix to the intermembrane space. Intermembrane space is where? It's between the two membranes. So this is your intermembrane space. So this is where we're pumping. So pump from the matrix to the intermembrane space. So we get a different color to make that. So therefore, H pluses are going from the matrix to the intermembrane space. They're going in that direction. So let me ask you now, where is the higher concentration of H pluses? Matrix or intermembrane space?
The matrix.
Pumping, going from low to high, it's going from here to here, so therefore your higher concentration must be in the intermembrane space, lower concentration in the matrix. So it's pumping, that's why it requires energy, all right? And so they're pumping H pluses from the matrix to the intermembrane space. Then what does it say next? H plus then moves back across the membrane, passing through channels in ATP synthase. So first of all, ATP synthase. Let's look at that. ATP synthase. ACE, what do we know about this? It's an enzyme. Look at so this is an enzyme. Look at the name, ATP synthase. What do you think this enzyme does?
Synthesizes
It
ATP.
synthesizes ATP, All right, so this is going to help make ATP. So look at here, H plus then moves back across the membrane. So we just used energy to pump H pluses in the intermembrane space. It's going to go back across the membrane. So where's the H pluses going? It's going back across the membrane, so it's going to go back to the matrix. It goes through channels in ATP synthase. ATP synthase is in the membrane, in the inner membrane. So in my little box there, that's the protein, and it goes back through. So what does this do? So something about the H pluses going back into the mitochondria, into the matrix of the mitochondria, changes the shape of this ATP synthase to the right shape so that it can phosphorylate and make ATP. So what does it do? It puts the phosphate back onto ADP to make ATP. So this, when we say this enzyme is going to make ATP, this is what it's doing. So remember, an enzyme has a specific shape, right? And the shape determines the function. The active site has to be the right shape in order for ADP and a phosphate to come in and this enzyme to put those two together. And something about the movement of the H pluses through that enzyme changes the shape of that enzyme so that it actually can put the phosphate back onto ADP. So this is what we call chemiosmosis, the use of energy. Here's another one, in an H plus gradient to drive cellular work. What's a gradient?
Concentration.
A difference in concentration across the space. So we talked about that with diffusion, like when I sprayed the perfume up here, or pretended like I sprayed the perfume up here. This was a high concentration, low concentration in the back. We would say that there's a concentration gradient across this space. So in this example here, we have a hydrogen ion gradient. Across what space? You have a high concentration in the intermembrane space, a low concentration in the matrix. So therefore, we have a concentration difference across the intermembrane. There's high and low. So I'm going to write that here. So this is a difference in, and I'll put the brackets, H plus ion concentration across the intermembrane. I think this is a good spot. I'll leave you in suspense about how it actually makes the ATP, and so we'll get into that tomorrow. So this is a good spot to leave off. I put up here that there's nothing due tomorrow. You have a review sheet for Chapter 9. If you haven't noticed, like with the Chapter 8 review sheet, it kind of goes right with our notes and stuff like that. It kind of goes right with it. So it's good practice, but you should also be reading. So to quote a very wise student, that reading actually helps, all right, understand the material, and it does. All right, the same wise student told me that the lecture made so much more sense to this person after they had read it beforehand, because then it all made sense. So, from random student A to you, some advice. All right.